To continue our discussion, so this is example. This is the cell membrane. So protein molecules are hydrolyzed by protease enzyme. Okay, and lipid bilayer is hydrolyzed by lipase, and then the chain fibrin or carbohydrate chain fibrin, are hydrolyzed by amylase enzyme. Okay, so surface tension, it is the property of liquids arising from unbalanced molecular cohesive forces at or near the surface, as a result of which the surface tends to contract and has properties resembling those of the stretched elastic membrane. Okay, usually surface tension happens, okay, when there is what we call a forces, okay, of attractions in the surface of the cell membrane. Okay, so based on our experiment, usually we have a bile atong up to, then we put margarine on the both slides, and then after that one we place bile plus the margarine plus, and the other one is soap solution. So usually based on the result class, ang bile dito amo to siya ang na-dissolve because of the presence of the proteins, which is known to be the theory of the surface tension. Okay, so you cannot really relate because you don't have hindi nyo nakita talaga sa experiment, so pasensya na. Okay, so test for the constituents of protein. So red litmus paper plus water turns blue when suspended in the mouth of the test tube containing albumin. So albumin solution, kung turns blue siya, that is basic. So filter paper plus lead subacetate, so presence ng black color that indicates the presence of sulfur. So yung filter paper mo maging black siya. Okay, so color reactions, so try to see also the videos for Millon's test. Okay, egg solution plus 1 drop of Millon's test, Millon's reagent, so reddish brown precipitate. Okay, sometimes ay, ba't ang tagal sir, hindi pa mag reddish brown siya. So try to boil for 10 minutes or 5 to 10 minutes, so that you can arrive at the positive result of reddish brown. Okay, so patients kasi iba namin magkakita dali-dali dahil ay sir wala pa man result, so try to wait. Okay, so ma-positive na siya as long may proteins. Next, xanthoprotic reactions. 1 ml egg albumin solution plus concentrated nitric acid, you heat, cool, and divide into two solutions. Yung one part you add ammonium hydroxide, the other one is standard, so maging light orange siya. So the protein test is performed adding nitric acid then sodium hydroxide to a substance that is a positive yellow or orange, so positive sa amino acid. Okay, so example, tyrosine plus nitric acid, it will form nitrotyrosine, yellow color, or tryptophan plus nitric acid, so mga orange siya, nitroptryptophan naman, plus yung phenylalanine same as nitrophenylalanine, having yellow or orange color. That indicates the presence of these amino acids during the xanthoprotic reactions. So Hopkins school test, okay, 1 ml of the egg albumin plus 1 ml Hopkins school reagent, incline the test tube. Okay, the purpose of inclining, okay, to come up with the junction between two layers or violet ring, indicates the positive for tryptophan. So this is a purple or a layer that is positive for tryptophan, that is Hopkins school test. Okay, or glycosylic test. Next, we have bioretest. So 1 ml egg solution plus 1 ml 10% sodium hydroxide plus copper sulfate, you add drop by drop, you come up with light blue or purple solution that indicates the presence of proteins. Okay, next, ninhydrin, 1 ml egg albumin plus 0.5% aqueous ninhydrin solution, you heat, cool, and then take note of the color result. Usually the color result is deep blue, purple blue, that indicates for the free amino acids when using ninhydrin test. So when heated with ninhydrin, these molecules give characteristic of deep purple blue color. Okay, so ninhydrin plus amino acids, so it will form hydridantine. Okay, then yung ninhydrin plus hydridantine plus ammonia, then it will lead to proline. Kung proline siya, yellow. Kung alpha amino acids, purple. So usually proline, yellow siya. So indicates the presence of proline in ninhydrin solution. So kung purple siya, amino acid siya, we cannot know what is the amino acid. Basta amino acid lang siya. Either alanine, lysine, but if you add ammonia, it turns into yellow. So positive for proline. Okay? It indicates that the sample or the sample of proteins that is being tested in the lab contains a lot of proline kung may yellow. Pero kung blue siya, that is only for good for amino acids. Okay? Next, precipitation reactions. Influence of minerals and organic acids. So, yung egg albumin mo for sulfuric acid, white coagulation. White precipitate, this is coagulation. Coagulate siya o aggregate. Okay? 1 ml albumin plus concentrated HCl, egg albumin contains protein. So, when hydrochloric acid is added to albumin, it denaturates the protein. So, mag-dissolve siya. In the lab, the observations were as follow. The solutions, which initially gel-like, turns into watery and transparent due to the presence of hydrochloric acid. Okay? 1 ml egg albumin plus concentrated nitric acid. Take note, separation of two liquids.